I'll be discussing protein sequences and specifically point mutations. So for the DNA strands, the template strand was the top strand in the prompt, and the coding strand was the bottom strand. So originally it was 3 to 5 prime end. We cannot sequence it in a 3 to 5 prime end, so we had to flip it around to 5 to 3 prime end. And then we found that the beginning coding strand, the start codon was AUG, or in the coding sequence it would be AUG. So I switched it to mRNA with the uracil instead of thymine, and then placed the codons in for that, and that made it for amino acids because the stop codon does not count as an amino acid. So point mutations, they are results in the substitution of one base for another. So transitions are pyrimidines to pyrimidines or purines to purines, and transversions would be purines to pyrimidines. So a transversion would be adenine to cytosine, or guanine to thymine, or guanine to cytosine, and a transition would be adenine to guanine or cytosine to thymine. So a missense mutation is a change in the amino acid that may alter the protein function, or our protein is non-functional, so the wild type mRNA and the codons, and then the mutated RNA. So I took what would be valine and changed it to alanine. So this would be a transition because the thymine, or the uracil in this case, would be changed to cytosine. So you can see that with this, the valine, we changed the amino acids now as alanine. And a nonsense mutation, we would change the codon so it becomes a stop codon. So we changed AAG, which we changed that to UAG, which would become a stop codon. And this is a transversion because adenine to thymine, or in this case uracil. So we changed lysine to a stop codon, so the codon becomes a stop codon, so it deletes a codon. A silent mutation, it codes for the same amino acid, so in this case we just changed GUC to GUA, so valine still codes for valine. It's a transversion because cytosine is now adenine. So valine codes for the same amino acid. Read the mutation, stop codon is changed to a codon that codes for the amino acid, results in a longer protein. So basically the stop codon is not deleted, it's just changed so the protein can continue sequencing. This is a transversion because adenine changes to thymine, or in this case uracil. So the stop codon changed and allowed for 3 additional amino acids to continue the protein sequence. Here are my citations.